So the macromolecules, organic macromolecules containing carbon, we kind of memorize those. You have your carbohydrates, your lipids, your proteins, and your nucleic acids. So here they are. Carbohydrates, energy, think glucose. Lipids, think a couple of these are really important, but I think the one we're going to need to know later or sooner than later is the phospholipids. And this is what our cellular membranes are made out of. Phosphate and lipids. The phosphate is polar, but the lipids are nonpolar. The phosphates are hydrophilic, but the lipids are hydrophobic. So those are important. And also the steroids, remember, the hormones, cholesterol, vitamin D, and your triglycerides, which are a glycerol with 3 fatty acids. Proteins, think amino acids, think structure. Think of how our DNA builds our bodies based on our genetics. And the machinery of DNA, the transcription, and then translation to the ribosome to build proteins. That's what DNA does. It builds proteins based on amino acids that are pre-templated. And our nucleic acids, remember, ATP is a nucleic acid. And of course, deoxyribose nucleic acid and ribose nucleic acid. So the breakdown product of a nucleic acid is going to be called what's called a nucleotide. And that consists of a nitrogenous base like ATCG, adenine, thymine, cytosine, guanine. So a base that's nitrogenous. A sugar, which is always ribose. And a phosphate. That's why ATP is a nucleic acid. It's really one nucleotide. So nucleotides are the breakdown, smaller parts of the nucleic acids, ATP, DNA, and RNA. And that's where we're going to live. Right? So we kind of talked about what monomers were and what polymers were. So polymer is a polysaccharide or a protein or polypeptide, which polypeptide and protein are basically the same thing because they're chains of amino acids. A polymer is a triglyceride because there's 3 fatty acids in glycerol. And DNA, of course, has many nucleotides within its system, and RNA as well. So we'll talk about the difference between DNA and RNA and what transcription is and what translation is. Now the monomers are your monosaccharides, your amino acids, your fatty acids, and your nucleotides. And that's pretty good. Hopefully there's a nice table that'll help you memorize those and understand it. So here's the anabolic reactions, and they all lose a water. So that's anabolic. And then you have the things that are broken down into smaller parts, and that adds a water. So that's hydrolysis reactions, and that's catabolic. So it's all about building these macromolecules within your cell. So anabolic is usually endergonic. So that needs energy to put into it. Catabolic is usually releasing energy, exergonic, if that helps you. Okay, so if you add these two together, anabolic plus catabolic, you have the sum of all chemical reactions, which is metabolism. Straight up. So you can see a lot of these pictures. Pearson does a decent job showing you the general dehydration synthesis. So if you add 2 monomers, a water is lost in the reaction. So the OH here, and you don't have to know the chemistry of this really, you just have to know about the dehydration part. And you build up a polymer. This is only 2, so if this was 2 monosaccharides, this would be a disaccharide. This is one molecule. Notice I'm saying molecule now because we're getting large molecules, macromolecules. Hydrolysis is where you take a larger, or like a disaccharide or a dipeptide, you add water, and then you get two separate monomer molecules, one molecule. Like if this is sucrose here, then you break that down to glucose and fructose. Pretty good. And of course, it's reversible. As an example reactions, here's glucose and fructose. Two separate. So going in from left to right, going from left to right in this reaction. So if we go from left to right, is that anabolic or catabolic? Anybody with me? So we're going from one monomer plus one monomer equals a polymer. That's anabolic, right? So if it goes in that direction, are we going to use dehydration synthesis or hydrolysis? The easy way to remember is because we're going anabolic, we're building something bigger, it's synthesis. And synthesis always involves dehydration. That's how I remember it. Now, if we go the other way, if we go right to left, then we're taking a disaccharide, in this case, and we're breaking it down to a smaller, or two smaller monomers. So a polymer to a monomer, or disaccharide to two monosaccharides in this case. And that is water being used and added. So that's hydrolysis. We're hydrolyzing, where lysing means to break. So that's another way you could kind of remember those two, because they always get confusing for everybody. I'm not going to kid you. So carbohydrates, like I said, another name for sugars and starches. So CHO, like I said, it is just like, once I see that, that's carbohydrate. Two hydrogens to one oxygen, always. So the monosaccharide is the simplest and the smallest version, and the smallest unit of a carbohydrate is a monosaccharide. And our famous one is glucose. Another example is fructose. And another example is galactose. And we can go to another one called ribose. They're all monomers. They're all monosaccharides. Ribose is the one we see in the nucleic acid nucleotides, and deoxyribose in DNA. So deoxyribose in DNA just removes one oxygen, deoxyribose. So again, when you're at Chipotle later, and you're trying to impress the people behind the chips department there with the salsa, you tell them about DNA and RNA and what it means. So disaccharides are two sugars bound. So disaccharide has to be built up through dehydration synthesis by using two monosaccharides. Makes sense, right? And then two or more is a polysaccharide. What's our main polysaccharide in our bodies and as animals? Glycogen. That's the animal storage of glucose, the polysaccharide glycogen. Now in in plants, it's called cellulose. Cellulose, remember, that's a polysaccharide in plants. And this is insoluble. This is our dietary fiber. We can't break this down. We eat this for fiber. Can't bust it up. We don't have the enzymes to do that. So there we go. So again, general formula. How many carbons, how many hydrogen, how many oxygen, like glucose? C6H12O6. I wonder how many times I say glucose in 2.5 hours. So again, this is the simplest of it. It's a monosaccharide. It could be smaller. You could have 5 carbon sugar, like ribose is a 5 carbon sugar, or pentose, right? And deoxyribose is removing one oxygen from ribose. Very similar. So it looks like this. Not that it's going to be, any different looking, but it's kind of like a house looking, where hexose is more like this. Like a razor blade, I guess. But there are like some 6 carbon sugars that look like this because they just add a carbon up here. But ribose kind of looks like that, 5-sided. And glucose is six-sided, hence the hexose. And fructose is the same too. So they kind of look similar. You know, this one looks more like the house. It's a pentose. This is a no. This is a hexose, just that the carbon is here. Right, that's the difference. So glucose is a 6-carbon sugar. Galactose, 6-carbon sugar. Deoxyribose and ribose are 5-carbon sugars. So this one is actually a 6-carbon sugar. If you need to know that. If you want to know that. But the main thing is that these are all monosaccharides, the smallest unit of a carbohydrate, right? So then you have disaccharides, which are 2 sugars bound together, 2 monosaccharides. Sugar is just so easy to say. So sucrose is glucose and fructose. Maltose is glucose plus glucose, that's 2 glucose. And lactose is glucose and galactose. So there we are. And here's let me ask you, see if you can remember this. This is taking 2 monosaccharides, making a disaccharide. So you lose a water in that process out of this reaction. So that's a dehydration synthesis, going from smaller units, adding them together to get one big one. Dehydration synthesis. So here's some of the disaccharides and what they look like. Sucrose is glucose and fructose. Maltose is 2 glucose, and lactose is galactose and glucose. You've heard of lactose intolerance, right? And usually the problem with lactose intolerance, especially genetic version, is they don't have the enzyme that breaks up lactose, and that enzyme is called lactase. And lactase is a protein that has to be transcribed from DNA to RNA, and then the protein is built on the ribosome with the help of messenger RNA and transfer RNA. So that's a genetic problem, that you can't make this protein lactase that splits lactose and lowers the energy of activation to make it happen faster. So it's a very uncomfortable thing when we're at Chipotle and we're eating the cheese, or if we get a chocolate milk or something, if they have chocolate milk at Chipotle, I'm not sure. Okay, now polysaccharides, polymers. So polymer is just a general term for many parts. A monomer is just one, as you've seen over and over again. So that's formed by dehydration synthesis. You're synthesizing something bigger from smaller. So here's some important polysaccharides. Starch we get from plants, which we can digest. That's soluble. That's the good stuff. Like a potato is a typical starch, rice and wheat grains, breads, and pasta, I guess, right? That's carbohydrate in a plant. That's how they store it. We store it in the form of glycogen. 2 different things. And chemically, they're different as far as how they're set up. Like glycogen is more branched chains, where starch is pretty straightforward. So glycogen is not very soluble. All right. Cellulose is insoluble. Makes sense. You look at glycogen is very branched. And this is just showing you, the drawing of it, the cartoon version of all, and this is all glucoses bound together. Every time 1 glucose is added, you lose a water. This is 4 glucoses added here, you lost 4 H2Os, 3 on this one, and on and on and on. So glucose is our storage of, glycogen is our storage of glucose. So when our blood sugar goes low, and a hormone called glucagon is going to be released, and we're going to break down glycogen, split it up to increase our actual blood glucose. Because you need to be homeostatic, right? 90 to 110 milligrams of glucose per deciliter. So lipids, moving along, lipids also contain CH and O, but it's not a 2-to-1 ratio with hydrogen to carbon. So these are not happy with water. These are hydrophobic. They're insoluble. They don't meld into water. They're hydrophobic, whereas a carbohydrate is extremely hydrophilic. So water phobic, to fear. Right? And here's our types. We have the triglycerides, which are stored in our adipose tissue for stored energy, actually long-term energy. phospholipids we got to talk a lot about. I don't know why this is uppercase, that first H. Looks like PH, but no. Here too. I'm not sure why that does that. No reason. I can't think of any reason. So phospholipids are important for our cell membranes, plasma membranes. Hopefully we'll talk about that in a little bit if we're not too tired. Really interesting. Steroids, of course, mostly those hormones, all based on what? Based on cholesterol. That's the precursor molecule. Eicosanoids are also lipids. They're like short fatty acid chains. And we're not going to talk about them a lot here because I like to talk about eicosanoids that are used as hormones. And an example of that is something called a prostaglandin. Prostaglandin has a lot to do with inflammation, which is really a big problem in our body. It's completely appropriate when you need it. You need prostaglandins. In many parts of our body, it's important for a lot of different things, like vasodilation and uterine contractions and other things, and inflammation when it's appropriate. But sometimes this can get out of control, and you have to take something like an Advil or a leave that kind of cuts down on the prostaglandins. That relieves pain, because too much prostaglandin release can lead to pain. If any of the Anthonys ever had a menstrual cycle, they know that it can get cramps and it causes pain and inflammation. So that comes from release of prostaglandins and secondary to major hormonal changes that happen to us once a month. So I'm not going to talk a lot about prostaglandins, although I like to, but that's more, to me, that's more in the endocrine realm, like a local release of hormones. So triglycerides are probably the one you should know the best. They're called the fats, solid fats, and the oils when they're liquid at room temperature. And these are made up of smaller parts called fatty acids. And there's carbon bound with hydrogen. Like you may have heard of like a saturated fat, like a saturated fat or a monounsaturated fat. So what we're talking about is these carbon chains, and what are these fats saturated with? They're saturated with hydrogen. So the carbon chains are saturated. Every carbon is bound to a hydrogen. And if it's monounsaturated, like I think olive oil would be monounsaturated, then one of the carbons is not saturated with hydrogen. It has a double bond and there's one less hydrogen. They share 2 pairs of electrons instead of 1, like usual. And then there's polyunsaturated fats, like vegetable oils and things like that. So it's hydrocarbons, long chains of carbon bound to many hydrogens. And there might be a picture coming up. So triglycerides have been around for a long time, obviously, when we really didn't have enough glucose for energy. So I don't think evolution caught up to this yet. I think we really don't need as much adipose stored for energy as we used to, right? And it could be because we just eat too many carbohydrates. So it's never used and it just becomes, fatty tissue within the cells. So the energy storage part, of course, is important for when you have no carbohydrates, but it's a secondary storage for long term. So these 2 things are really important: insulation of the organs to keep the temperatures correct, and protection, because remember your abdomen have no bones in front of them, right? There's nothing there. Like, I don't know if I was telling you guys, if you wanted to kick somebody and hurt them, you don't have to kick them in the chest because the sternum is there. You're not going to kick them in the head because there's bones there, there's cranial bones. But kick them in the stomach, there's really no protection besides this adipose curtain in front of there. And everybody has it, no matter how thick they are in the middle. Okay? But there's another function of adipose tissue, which is mostly triglycerides. I'm talking about storage of triglycerides in adipose tissue, which is connective tissue, is hormone release. Your adipose can release hormones. And they're important for reproduction, and they're important for some brain functions as well. So the research is still going on about that, but still it's more than just energy storage these days. Okay, this is a glycerol with one fatty acid. So let's just jump to the triglyceride, which has three fatty acids. One of them is unsaturated, and the other two are saturated. It doesn't. You don't need to know that. Just know that there's a glycerol and three fatty acids. So there's yellow things, 1, 2, 3 fatty acids bound to a glycerol. So you should kind of know what that looks like compared to glucose, right? So far, here's another version of the way it looks. Probably should be seen in a slideshow. So run this through a slideshow on the static part, and you'll see how this kind of feeds into each other. Look at this one. Looks like they just threw this up here as an animation. This should be nice. I don't think I've seen this. So this is, again, the fatty acids, perfectly seen. This is a saturated fatty acid. And there's another one. Okay, and another one here. Fatty acid. And here's a glycerol molecule. Don't have to memorize anything about how to draw this. Please don't do that. Just know that the 3 fatty acids are, and a glycerol make up triglycerides, hence the term. So saturated fats are all carbons are linked to a hydrogen. So it's saturated with hydrogen. So this is kind of harder to break down, your animal fats. Butter is a good example of a saturated fat. I think the only vegetable saturated fat may be found in something like coconut, right? And you probably know another. So I can't think of one right now. But animal fats are saturated fats, meats, beefs, very saturated, which is probably and definitely, I should say, definitely worse for your cardiovascular system than a monounsaturated fat like olive oil or canola oil or vegetable oils, which are polyunsaturated. Okay, so saturated fats straight up, no double bonds. Now, ignore this oxygen double bond. The double bond would be down with the hydrogens. So don't worry about this right here. This is bad. Like unsaturated fats, again, are okay. You have few double bonds. And you might hear things like omega-3 fatty acid or omega-6 fatty acid. They're both good for you, for your cardiovascular system. And they're unsaturated fats, but the 3 and the 6 refer to which carbon there's a double bond on, which was not saturated with hydrogen. So from what I know, the omega-3s are probably better because they don't increase inflammation, where omega-6 is good for your lowering cholesterol and keeping your triglycerides under control. And it's good overall, but this may add more inflammation where omega-3 doesn't. Trans fats are definitely not good. This is something made in a laboratory to kind of, trans is just the shape of the fat. They actually manipulate it to kind of resemble a fat, and they use it to keep the shelf life a little bit longer, or maybe make the food a little bit more, the texture of the food a little bit better. So this is really dangerous for your cardiovascular system and your blood vessels. So that it is considered unhealthy. There's no question about it. Trans fats. So you want to keep that low. And if you read packages, sometimes it'll say 0 trans fats. But even less than 0, right? They'll just say 0 trans fats. But sometimes it's misleading, because if it can be less than 0, what if there's 0.9? That's less than 0, but that's still trans fat. So you got to be careful with that kind of packaging. So I think like the book says here, I think they just mentioned omega-3, because that might be the healthiest. And it really is healthy. Yes.
I don't think 0.9 is less than 0. It's more than 0.
Yeah, I'm sorry. Like anything less than 0. Sorry. Yeah, like
less than 0.
Sorry. Right. Good point. Who's that? Emily. You're always on it, right? Right. So whatever, something lower than 0, right? Which could be even a trace of it. So that's kind of a trick I read about these packaging that goes on, where they could say it's 0, but even a trace of it, like a fraction, is even too much for trans fats. Even if they use it in, and now McDonald's, of course, is I don't know about Chipotle, but McDonald's really had to come clean with a lot of that stuff. So again, it's not a nutrition class, but you should understand the way the fats work and which are better. So this is right here is a monounsaturated fat. But this is the triglycerides. So 1, 2, 3. Oils, fats at room temperature. So here's the important thing that we should get this down now, phospholipids. So modified triglycerides, where you have a phosphate, which is polar. The phosphate head is polar. And the tail is the lipid. And that is the non-polar. So the membrane is going to be a biphospholipid layer. So it kind of looks like this. It looks like a balloon with 2 tails. And it's 2 layers. It's inside and outside aligned with the phosphate. And it has the lipids in between. So it kind of looks like this. So the outer and inner part is phosphate, these balloons. Like if this was the cytosol inside the cell. And outside the cell would be the ECF, the extracellular fluid. So the phosphate faces that part, and the inner part here is your lipids. Fatty acids, 2 fatty acids attached. So that's the 2 strings to the balloon. So the inner part is hydrophobic, and the phosphates are hydrophilic. But I have to tell you, I think the membrane, for the most part, is hydrophobic. So it's easier for lipid-soluble substances to get through this membrane, and that's called permeability, which we'll talk about again. That's a really important concept. Okay, here it is, phosphate head, and it's just showing you the chemical structure and this. So we really don't have to worry about drawing any of this, but again, you should know that the way it looks as far as that balloon and phosphate head and the tails, because you're going to see this many times over. And this is telling you about the polar versus non-polar. That's really important that you get that down. Here it is. So this is a good way to go into this. So this is what the cell membrane looks like, a biphospholipid layer. So each one of these here looks like this. Non-polar tails, polar heads. The heads are the phosphate, the tails are the lipids, which are 2 fatty acid chains. Steroids. Now this is based on cholesterol as the precursor molecule for all steroids. Okay, and cholesterol also is found in dairy products, right, and animal products. And vitamin D, actually, is based on the steroid model. Steroid hormones, we mentioned these a lot: estrogen, progesterone, testosterone, aldosterone, cortisol. All steroids, or cortisone is an example. The glucocorticoid, again, we don't do endocrine in this class. So bile salts, bile, we'll talk a little bit about that. Bile is an emulsifier released from the gallbladder. And you should know, like in lab, you should know where the gallbladder is and where the liver is. So bile is actually made in the liver. And bile salts, or bile, is an emulsifier, not an enzyme. So what it does is it kind of takes the fats that you eat. And Chipotle, we can get some of those. And the gallbladder will release the bile, even though it's made in the liver, and try to make the fats a little bit more hydrophilic and a little bit more polar, so they're easier to break down by the protein enzymes later on in the digestive system. So that's important in digestion, actually, and that's about it for bile salts. They can be converted back to other compounds in the liver. So the liver is a manufacturing machine, really, but it's part of a few different systems. I think we kind of talk about that in lab and a little bit in lecture. So another thing, and remember this, cholesterol, besides, being used to make steroids, it also is embedded in the cell membrane. And for no other reason except to give it a little structure and support. So this is one time where lipid is being used for strength and support. So cholesterol is part of the membrane structure. So remember that for later, because that's important. Here's what it looks like. 3 6-carbon rings and 1 5-carbon ring. So you don't really have to, again, this is the part that changes between different structures. Or cholesterol, this is cholesterol. So if you add different structures up here, you'll get testosterone, estrogen, very similar. In fact, your body can convert estrogen to testosterone if it has to, or testosterone to estrogen if it has to. So again, that's more of an endocrine. So here's eicosanoids, and they're derived from these fatty acid chains, usually about 20 fatty acids, and a chemical or compound called arachidonic acid. So arachidonic acid makes these eicosanoids. So prostaglandins, I mentioned before, important for blood clotting and inflammatory labor contractions, but production of prostaglandins can actually be a problem and cause a problem in homeostasis. So prostaglandins causing pain, if you take some NSAIDs, which are non-steroidal anti-NSAIDs and inflammatory drugs like ibuprofen, block the enzyme that builds prostaglandins, like aspirin too. That's acetylsalicylic acid. Ibuprofen is Motrin and Advil, and the other one is naproxen, are all in the NSAID group. Acetylsalicylic acid is aspirin, ibuprofen, Advil, and Motrin. Naproxen is Aleve. They're all NSAIDs, non-steroidal. So the main steroidal anti-inflammatory steroid is cortisone, which is a glucocorticoid. Again, we'll talk about that in the nervous system, but it's really endocrine. So I would love to talk about that all day, but again, we got to talk about the skeletal system and the cell first. So proteins, a big part of the structure of the cell, 20 to 30%. So structure, enzymes, mostly protein. And of course, you have contractile proteins in muscles. Like you're going to hear of these two a lot in a couple of weeks. And that's one of them is called actin and myosin. And those are contractile proteins. You also have structural proteins within muscle as well. So they contain the CHNO, but now we're getting more into nitrogen. Amino acids carry nitrogen more than carbohydrates and lipids. And of course, nucleic acids carry nitrogen as well. Some sulfur, some phosphorus. So we're really concerned about CHON, and we're concerned that protein is a macromolecule that's organic, and it is used for our structure. I know this, very important. So amino acids are the monomers, and they're held together by covalent bonds. Make no mistake, these are covalent bonds. We just call them peptide bonds. And the shape of the protein is very important. And the different levels, you're going to have primary shape, secondary shape, tertiary shape. How do you even spell that? And quaternary. And really, this one right here, the tertiary shape, is probably the one we deal with the most because that's the real functional specific protein shape. And that, again, has to be designed through the machinery of DNA being transcribed to RNA, messenger RNA, and then sent to the ribosome to translate that into proteins like an enzyme. So structural proteins, again, you're going to hear this term collagen a lot with connective tissue, but also in muscles, because collagen is the most abundant protein in your body, and connective tissue is the most abundant tissue in your body. So collagen is a big protein, very important to make that in abundance. And enzymes are specific for certain things, which we call substrates, like a disaccharide is a substrate for the enzyme. So we'll say this, like substrate is a general term for what the enzyme is going to work on. So substrate. So let's say the disaccharide is lactose. The enzyme, and the enzyme would be lactase, specific protein that's specific for lactose. And lactose is the substrate, the disaccharide. So it could break it up into smaller monosaccharides and then and it's much easier, it's a lot easier to reabsorb or absorb the monosaccharides in our large intestine. And here's an example of a quaternary structure now, it's the only quaternary structure of protein we'd see, and it's called hemoglobin, because it has 4 polypeptide chains. And by the way, all those peptide chains are held together by hydrogen bonds. It seems so long ago I talked about that. So hemoglobin is a carrier for oxygen. So in one hemoglobin molecule, which is 4 polypeptide chains, you can carry 4 molecules of O2, of oxygen. And there's 250 million molecules of hemoglobin in one red blood cell. And I couldn't even tell you how many red blood cells we have. You'll never have to count that here anyway. This is an example of actin and myosin, the way they swing together, and we'll do a lot of this. Right here, what you're seeing right here, we got to talk a lot about soon. So they're proteins that contract, shorten segments of muscle. So that's all muscles really do. They contract. That's all they do. They shorten. Then you have communication proteins like receptors. And look at the membrane. Here's the cell membrane. And then you have an embedded receptor for something like insulin, say. So insulin will bind to the specific protein receptor within the membrane and cause a change. Like in insulin's case, it'll facilitate the movement of glucose into the cell, so it could be used for energy. Then you have your defense proteins, or your blood antibodies, are mostly protein. And the antigens are also protein too, which you'll see in AMP2. So again, back to another repetition, amino acids bound together through these covalent bonds, which in proteins we call peptide bonds. So there's 20 types of amino acids. Sometimes we say essential amino acid. If we say essential amino acid, I think there's like 9 of them. Essential means we have to get them from our diet. We don't make them within our own bodies in the ribosome or in the rough endoplasmic reticulum. So you can probably get those at Chipotle. You get the right mix of beans and rice. You don't have to have any meat. And you could probably get those essential amino acids and build good proteins. So it's good to be vegan if you are. And you can do it. So again, these are covalent, make no mistake there. So they have an amine group, of course, and an acid group. And there's a functional group called the R group that is the only thing that makes each amino acid different. They all have amine groups, obviously, and all have acid. That's why they call it amino acids. And there it is. So they all have this. This is the nitrogen-containing part of the amino acid. And there's the acid. And this is what changes with the 20 different amino acids. Didn't you just see that? So here we go again with the monomers, 2 amino acids put together, making one dipeptide. That's what we call 2 amino acids bound together. And if we go from left to right, that's an anabolic reaction. We lose a water. That's dehydration synthesis. If we take a dipeptide or even a protein or a polypeptide and we break it down, so every peptide bond that's broke, you have to add a water. Every peptide bond that's put together, you lose a water. So this is, again, an example of dehydration synthesis, anabolic, and this way is hydrolysis. And that's where you need to live in this whole chemistry business. All right. So primary structures of protein is usually just a peptides bond together, like it could be straight across. Your book may draw them square or rectangular. Secondary structures, again, a little bit more mature, if you will, or advanced protein being built that has a shape like an alpha helix or a pleated sheet. And that's what it looks like, a pleated shades. Alpha helix is like that coiling DNA, like a spiral staircase. So that's, remember, these two are secondary structures, and hopefully there's a nice picture coming up for you. Tertiary structures are now more of the functional shape. So that's even further on in the progression of the maturity of a protein. And these are important for enzymes. They have to be very specific, right? Specific enzyme for lactose, specific enzyme for sucrose to break that down. And those are just the digestive ones. And there's so many, I can't count how many enzymes you have for all these reactions. But most of them are protein. Coordinary, the only one with the 4 polypeptides is hemoglobin. So let's break down this word. And this is the molecule of hemoglobin. Basically, that's a protein, carries oxygen. Heme is, again, refers to blood, but the heme portion contains iron. So you need iron within that hemoglobin molecule. 4, actually, there's 4 hemes. The globin is the protein or polypeptide. So the heme is where the O2 binds with the help of iron as a cofactor. And there's 4 binding sites for oxygen on one hemoglobin molecule. It's 4 polypeptide chain. So the shape of hemoglobin is extremely important. So if you have a genetic problem with the machinery, again, that builds hemoglobin, like something horrible like sickle cell anemia, which is a change in the shape where you can't really carry oxygen. You can't even get the cell through a capillary, right? So hemoglobin shape, extremely important. The shape of muscle cells, everything is important. So, but hemoglobin is the only example we're going to use for a coordinary shape. So the primary structure is kind of straight across, amino acids being peptide bonds, covalent bonds in a row like this. This is your secondary shape, which is like a helix. And this is a pleated sheet, if you will, kind of folds on each other as it's developing. Tertiary structure now is very specific. Now this is where the enzymes or even receptors have to be very specific for their substrates. Or we'll hear the word ligand, which we'll talk about for hormones. So this is really important. The coordinary, basically, this is what hemoglobin looks like. And when you know, again, we don't really do hemoglobin in this semester, but you have to know what blood tissue looks like. Blood is a connective tissue. So it's important. Here's a protein structure animation. Very good. Okay, you can have fibrous proteins, and those are the ones that kind of make up the cytoskeleton of the cell. And the globular ones are more like your antibody type proteins. Strand-like, not soluble in water, there's a protein called fibrin, which is helpful in blood clotting, but it's insoluble, so it's kind of a problem, especially with positive feedback, right? We talked about that last time. And most have these tertiary shapes, some coordinary, but we're only going to mention hemoglobin with that this semester or next semester. So other examples of fibrous are your keratin, which is the protein that gives skin its strength, elastin, which is found in many connective tissues that give any connective tissue that needs to have elasticity. Collagen is what gives it the strength, though. And then certain contractile fibers, don't worry about that. We'll talk about actin and myosin when we get there. So here's your globular proteins. And now not only antibodies, but the protein hormones, like insulin again, and growth hormone, anything that's not a lipid hormone. And mostly tertiary structures, very specific. Okay, you don't have to worry about molecular chaperones, but enzymes, that's really important. Enzyme, very specific. The structure is extremely important. Okay, denaturation means you have a hostile environment that is going to destroy the structure of a protein. It's going to affect the hydrogen bonds, actually, because hydrogen bonds are what keep that functional 3D shape together. Okay, so like there's a protein in eggs, right? It's called albumin. So did you ever fry an egg? Like where you, oh, here it is. Here we go, cooking an egg. Like when you, like say you crack an egg and you just want to eat the egg whites. You know, you put the egg whites into a bowl, you get rid of the yolk. So the egg whites, before you add the heat, are white. It's albumin, the protein still maintaining its shape. But if you put it under high temperature, which is a hostile environment, it's going to destroy and denature that protein albumin. It's going to turn white, to show you that. So increased temperature is a hostile environment for proteins in the body. And acidity, decreased pH, is a hostile environment for your proteins, except in the stomach. Now, the stomach is a place where there's excessive HCl, hydrochloric acid, and there's proteins that are enzymes that could live in the stomach because that's the way they evolved. But there's nowhere else in the body that has a low pH, like a pH that's like below 3. I mean, occasionally the vagina will go down to a lower pH, but not close to 0 like the stomach. So there's enzymes in there that actually work that way. Let's turn this off. Sorry about the noise. Okay. So the shape is very important. The environment is very important. So enzymes, protein, shape, specific, specificity, tertiary shape, sometimes quaternary, right? So enzymes are catalysts where they speed up a reaction. They don't just make the reaction, they don't push the reaction. They decrease, enzymes, these catalysts decrease the energy of activation for that particular reaction to go forward. So that speeds it up. Okay. And you're not going to have to know all of this, you know, what the difference between a whole enzyme and an apoenzyme. You don't need to know that. Sometimes you need a cofactor, like something like iron acts as a cofactor. And a coenzyme could be a vitamin to help the enzyme. So a lot of research still going on about certain vitamins that are good for enzymatic function. You should know what a substrate is because the enzyme acts on the substrate, or the substrate and the enzyme are specific for each other. Like lactose is the substrate for the enzyme lactase. That's the best example for you. Usually ends in ase, but not always. Like there's an enzyme in your stomach called pepsin, and there's an enzyme from your pancreas called trypsin. And they're both proteins. Pepsin breaks down proteins in the stomach. That's the one with the acid. Trypsin also breaks down protein outside the stomach, released from the pancreas, exocrine, digestive enzymes. So enzymes lower your activation energy. So that's the energy to get this thing started, prime the reaction, and off it goes. So like I said before, like a reaction that takes 0.5 seconds to go through with an enzyme could take hours if the enzyme is not present or it's not active, or it's inhibited by a medication or genetics or a hostile environment where it's denatured. So the substrate binds to the enzyme and the active site, of course, and it forms a complex. And then it rearranges, and the final product is the action of the enzyme, whatever the action is going to be. And that could be breaking things up, anabolic, the hydrolysis, or it could be building things up through synthesis, like dehydration synthesis, anabolic, catabolic versus anabolic. So this is just showing you the energy. Without an enzyme, it's high. It's going to take longer to prime this reaction. So this is the activation energy right here is lowered with an enzyme. So remember and that's the question, the enzyme lowers the activation energy that's required to start a reaction and continue a reaction. This is probably a good slideshow to go through, looking at the enzyme. And here's the substrates, amino acids. This is an anabolic reaction and forming the bond between speeding up this dehydration synthesis. And off it goes at the end. You have yourself a dipeptide with a dehydration synthesis with the help of an enzyme. It could happen without, but it's not going to be as fast, and it's going to take more energy that maybe your body can't afford. Right? So this kind of goes through a slideshow showing you this dehydration synthesis reaction forming your dipeptides. Okay. So I think if I asked you, give me an example of a protein, you're going to say enzyme, I hope. Okay, nucleic acids. Now, same thing. CHON is CHO, but now you have nitrogen and phosphate, kind of like the proteins. And these are the largest molecules. These are really long, these nucleic acids, especially DNA. So the polymers are the DNA, RNA, and don't forget ATP, but it's just one nucleotide. So the breakdown products, or the monomers of nucleic acids, are nucleotides. And that's a nitrogenous base, A, T, C, G, and uracil for RNA. So adenine, thymine, cytosine, guanine, and uracil. So you have a pentose sugar, whether it's ribose or deoxyribose, and a phosphate group. There's those big H's again. What's going on there? Okay, so deoxyribonucleic acid and a ribonucleic acid, RNA and DNA. Remember this, DNA never leaves the nucleus of a cell. Never leaves, stays in the nucleus, DNA. There's three types of RNA, and there's mRNA, which is messenger RNA. Then there's tRNA, which is transfer RNA. And then there's rRNA, which is ribosomal RNA. So the messenger pretty much does what it says. It's a messenger. It takes the code from DNA and leaves the nucleus with it. So going from DNA to messenger RNA is called transcription, and that happens in the nucleus. Again, that's in nucleus. Messenger RNA leaves the nucleus and then builds proteins at a place called the ribosome. That's the site, that's an organelle, that's the site of protein synthesis. So transfer RNA brings in amino acids to the ribosome. Messenger RNA, ribosomal RNA is basically building ribosomes. So, and we'll go through that when we do other parts of the cell. So these are the nucleotides. Again, you have a base, sugar, and a phosphate. Sugar is deoxyribose or ribose. So now this is DNA. So first of all, DNA is double-stranded, or RNA is only single-stranded helix. And these helixes, it's like a ladder, and the sides of the ladder are the actual sugar and the phosphate. And the bases form hydrogen bonds between the two strands. So phosphate group, of course. And then the nitrogenous bases of DNA are ATCG, adenine, guanine, cytosine, and thymine. So the purines are a little bit bigger, maybe one extra carbon ring. And the pyrimidines, cytosine and thymine, are a little bit smaller. So they pair, in the DNA, they pair A to T and C to G. Now, you're going to find there's no thymine in RNA. Thymine is replaced with uracil. That's another difference, I guess. So ATCG with those base pairings. And these are bound through hydrogen bonds. So it's kind of showing you this double-stranded DNA, ATCG, ATCG. And this is the pairings that's going to be transcribed onto messenger RNA that's going to set the template of pairings to make amino acids. And then the amino acids on the ribosome are going to build proteins. So DNA is transcribing proteins and it's translated at the ribosome. So translation happens at the ribosome. And we'll talk more about that in the cellular lecture. So RNA links DNA protein synthesis is slightly different. It's single-stranded, right? Ribose is the sugar, not deoxyribose. So instead of AT, it's AU, because uracil replaces thymine in RNA. Still CG or GC. Okay? And then you have your messenger RNA, your transfer RNA, and ribosomal RNA. Messenger RNA is the one that is transcribed in the nucleus. Transfer RNA brings in the amino acids in the right order. And ribosomal RNA basically is made of ribosomes and builds more ribosome RNA. So this is a really good animation. I like this. Genetics is tough. The whole genetic part of it is kind of tough. This is ATP, adenosine triphosphate, a nucleotide really, because it's got adenine. And it's got a ribose, and it's got phosphate. So technically ATP, this energy, this potential energy molecule is a nucleic acid. Really important. So we use glucose, and don't forget we use oxygen, and we use the mitochondria to make a whole boatload of this stuff. Adenosine triphosphate powers chemical reactions, not actually directly. It has to be hydrolyzed in a lot of cases, and it has to be synthesized in a lot of cases. So it's a lot of energy building compounds, ATP. Okay, it contains RNA nucleotide really, so that the sugar is ribose and has the phosphate. Again, you see 3 phosphates, nothing that you can't see. So there could be adenosine, which is with no phosphate, and adenosine monophosphate just has 1 phosphate bound, and adenosine diphosphate, 2 phosphates, and 3, the one we know and love, ATP. Okay, so that terminal phosphate can be transferred to other compounds, yes. And again, I don't think in this semester we're really going to talk about AMP. No, we don't. Maybe in AMP2 a little bit. But this conversion is important for us. As we talk about sodium, potassium, ATPase pumps. I know you don't know what that is right now, but you will. All right, here's a reaction. All right, and if we hydrolyze ATP, we're going to have to, of course, add water. It's catabolic. You get adenosine diphosphate plus 1 inorganic. The I stands for inorganic phosphate. And this is a boatload of energy right here. There's only 1 ATP, though. You need a muscle cell, I think, per 1 molecule of glucose could give you about 36 ATPs per 1 molecule of glucose. That's pretty efficient, like a skeletal muscle cell or cardiac muscle cell. Now, if we're making ATP, we're going to have to take a phosphate. All right, you're going to have to add the energy because it's endergonic. Dehydration synthesis to make the bigger molecule of ATP, adenosine triphosphate. Yeah, so, and the enzymes use this, enzymes are used in here, and you use, go from left to right, you use what's called ATPase. It's cleaving, it's lysing and hydrolyzing ATP into these 2 products, your ADP, adenosine diphosphate, and the inorganic phosphate. If you're building it, you use an enzyme and you lose water in this dehydration synthesis, you would use ATP synthase. Crazy stuff, man. I don't make this stuff up. And that's going to be important for some of the cell membrane things, like your transport, like your sodium potassium pumps, like the solutes or the sodium and potassium. Again, this is not showing that exactly, but ATP is in the membrane, and that could act as a channel to let things in and out. So the energy has to be readily available to move things against its gradient. So again, we're not going there tonight, but we're going to talk a lot about that. Transporting things through a membrane. Sometimes it easily goes in with no problem. It's free down its gradient. That's called diffusion. Sometimes it goes against its gradient, and that's active transport. So we're going to talk all about that when you guys are refreshed after a week of going through all this. So to contract muscles, you need ATP at a constant rate. Like, of course, skeletal muscle when you need it, which is often, and then cardiac muscle when you need it every second of your life. You need ATP to keep those muscles contracting. So you need oxygen to make it, and you need the nutrients like glucose. Hopefully glucose is what you're using. And of course, you need ATP for chemical reactions, phosphorylation, which we could talk a little bit about, but that's really not part of this class. So how are we doing? Okay?